halfway through, we've looked at the cell wall, and then we're now discussing the outer membrane of the gram-negative cell wall, right? So gram-negatives have a outer membrane, which is again a phospholipid, but it is quite different from the phospholipid that is found as your cytoplasmic membrane. So if gram-negatives have two membranes, one outer membrane and one cytoplasmic membrane, you'd want to compare and say both of them have are supposed to be um, selectively permeable. So is their permeability similar or not is what we talk about. So as such, if you're looking and comparing your outer membrane to the plasma membrane, your outer membrane is more permeable. And this is because of the presence of some porin proteins which are embedded in the membrane, and also some transporter proteins that are there that will allow the passage of certain molecules. So the porin proteins form channels through which small molecules up from about 600 to 700 Daltons can pass through. So these channels are formed because of these porin proteins. Now, E. coli has been studied and has 3 porin proteins that we know, and they are called OMP-C, OMP-F, and 4E. Now, what does OMP stand for? Does anybody recall from the previous classes?
Outer membrane protein.
So these are outer membrane proteins. So it's telling you this is a gram-negative function, which is in gram-negative only, the outer membrane. So C and F are OMP-C and OMP-F are non-specific as to what they allow, except for the size. Whilst 4E is a very specific specific channel or porin protein that is made only synthesized under phosphate limiting conditions. If you're growing the bacteria under phosphate limiting conditions, or in nature it is not finding enough phosphate, then it will create this channel 4E9, which is very specific for anions. So remember, for phosphate being limited now, they want to take in as much as they can and make this special channel to go to use it to collect all the phosphate they can from their environment. Of course, this pore size varies with species, so they are not constantly the same size pores for outer membrane proteins. And of course, the speed of diffusion as to how fast the material comes in is dependent on, of course, the concentration gradient. So concentration gradient will again be the demanding factor, or will be the limiting factor in terms of speed of diffusion. Okay. So larger molecules use the specific receptors OMP-A, which is a different one, larger receptors, whilst this OMP-A is the one that serves as a receptor for amino acids and peptides that have to be that have to be transported across the membrane, outer membrane. And also this OMP-A is the receptor for F-pilus also. The F-pilus is connected with the OMP-A. Now, if you have a diagrammatic representation, you can see that this is your porin trimer. That means 3 proteins are there to make a channel. And these are the green and the yellow and the blue are represented here as 3 different proteins. So this is a protein porin trimer. And here is your OMP-F. If you're looking at it, the green one, you're looking at it from the side, it looks like a web or a weave, and making the protein is assembled such in the membrane that certain size molecules can go through it. Now, osmotic protection is a very important function, you understand, and this osmotic protection is usually given to bacteria by their cell wall. So what is osmosis, first of all? Can anybody tell me what is osmosis? The process of osmosis?
Is it the diffusion of water?
It's a diffusion of water directed by what?
The concentration gradient from
the concentration gradient. Sure, it will always be the concentration gradient across the membrane will drive the water either inside the cell or out of the cell. And of course, these 2 concentration differences are noted as hypotonic or hypertonic environments, right? So when there's a hypotonic environment, the solute concentration outside the cell is less than the inside, so the water will move into the cell and the cell will swell. And if the cell wall is not there, then the cell will lyse. So actually, you know that your cell walls are giving osmotic protection to bacteria. If they have a cell wall, their cell cytoplasmic membrane comes all the way, absorbs some water, goes up to the where the cell wall is, and then it stops. Then it cannot take any more water. So actually, cell walls do protect bacteria from in hypotonic environment. So my conclusion is that if you put bacteria in pure water, no salt at all, it is hypotonic environment for them, and they do not lyse in hypotonic environment. Only living cells that do not have a rigid cell wall will lyse in hypotonic environments. So what would be isotonic for your bacteria is something that changes from different bacteria, but most of the bacteria that we deal with in or near our body or in our body, they are usually their isotonic environment is between 0.5 to 1% sodium chloride. And that is why you know that when we say saline solution, saline is 0.85% NaCl, and that is isotonic. So when you want to prepare any media for the growth of the bacteria, of course, you want to have isotonic environment, and you will always add at least a 0.5% sodium chloride in your media so that the bacteria can grow comfortably. Now, in an environment that solute concentration outside the cell is greater, water leaves the cell. And what is the consequence of water leaving the cell and the cytoplasm shrinking? That means that your membrane, the cytoplasmic membrane, moves along with the cytoplasm shrinkage, and the inside of the cell becomes dehydrated. So what do you think is the consequence of having a dehydrated cytoplasm? What do you think happens to the cell or its metabolic processes? You know that a lot of reactions inside the cell are dependent on having water there. So if there is no water or less water, of course, your metabolic processes will stop or slow down. So the cell stops to grow or to divide. So in hypertonic environment, this process where you lose water from the cell cytoplasm and your membrane comes with the cytoplasm shrinkage, the cell wall still remains rigid, and you can see the shape of the cell remains the same, but your metabolic processes have stopped. And this process of shrinkage of cytoplasm with the membrane, the word is used is plasmolysis. Now, this is a very, I would say, incorrect word that has been coined for it. It seems like there is a lysis or breakdown of the plasma, the cytoplasm, but this is the word you will use when you place bacteria under hypertonic environment. Plasmolysis occurs, water is lost from the cytoplasm, and metabolic processes eventually stop. So what is some of the evidence that we have that cell wall is protecting the cell from osmotic lysis? There are two compounds that you will come across, and they've been used extensively in research when microbiology was first instituted. And you know this enzyme called lysozyme. This lysozyme is the one that breaks the bond between NAM and NAG. You remember where NAM and NAG are? What are they? Can somebody recap for me? Anybody?
They make up the peptidoglycan.
They are the main disaccharide of the that is repeated again and again, and the bond between them is 1,4 bond, right? The bond that we described. So lysozyme is a specific enzyme that breaks down that backbone of the the disaccharide sugar in the cell wall of bacteria. Where is this lysozyme found? Where would I find lysozyme? Anybody? Yes? No idea?
Tears?
In your tears, in your saliva, in a lot of your secretions. This is supposed to be your innate immunity that you produce enzymes that are destructive for bacteria. So lysozyme, you know who discovered penicillin? The name of the scientist?
Fleming.
Alexander Fleming. Alexander Fleming, before he accidentally found penicillin, he had worked with lysozyme for 10 years. He was trying to get it into a state where we could use it as a antimicrobial agent outside, I mean, in tubes, and he was working with this and determined how it breaks down the cell wall of the bacteria. And accidentally he left his petri dishes with Staphylococcus aureus, and contaminant grew on it, which was a fungal contaminant of penicillium. And this penicillium actually produced this compound called penicillin, which diffused into the medium and kind of destroyed the colonies that were growing, that had already grown near near where the fungal contamination was. So as such, I would say trying to find something that would break the cell wall and use that as an antimicrobial agent using lysozyme as their model, he was very lucky to have found the real compound that became such an important discovery in all of microbiology. Penicillin actually it inhibits peptidoglycan synthesis. So a new peptidoglycan is not synthesized, meaning your cell walls cannot grow. If the cell wall cannot grow, the cell cannot elongate. And if the cell cannot elongate, of course, cell division will not occur. So penicillin happened to be right where he was trying to find something to break the cell wall using lysozyme. He stepped into a chemical compound that would even stop the synthesis of the peptidoglycan. So if the cells are treated with either of lysozyme or penicillin, they will, if you put them in a hypotonic environment, eventually their cell walls are becoming weak. And what will happen? In hypotonic environment, they will lyse. So definitely having hypotonic environment showing us that the cell wall was very important, or especially the peptidoglycan is very important in the maintenance of the cell against osmotic lysis, right? Now we have certain bacteria. Most bacteria will have peptidoglycan in their cell walls, and that protects them from osmotic lysis. But there are some that, such as, for example, your mycoplasma. Mycoplasma are naturally devoid of a cell wall, means they don't have a peptidoglycan, and so they are quite vulnerable in hypotonic environments. These mycoplasma, and there are others that do not produce a cell wall, mutants of some bacteria you can have that do not produce a cell wall. And in the mycoplasma also, we know that they have some protection from osmotic lysis because of some inserts in their plasma membrane. So their plasma cytoplasmic membrane helps them survive much better, even in certain hypotonic environments. So here is a kind of a little model for you to say this is a rod-shaped bacterium. You treat it with penicillin, the cell wall synthesis will stop, and eventually the cell wall will be destroyed, and your cell will come into a spherical shape because the shape of the bacterium is maintained by the cell wall. So if the cell wall has dissolved, now you have something called a protoplast. The protoplast is cells that are devoid of a cell wall. And then when you transfer it into a dilute medium where water, hypotonic environment, so water will go in, the swelling of the cell will occur, and eventually it will lyse. So we can use penicillin or lysozyme, either of them, to show that osmotic resistance, resistant to osmotic lysis, is because of the cell wall. Archaeal cell, we'll talk a little bit about archaeal cell. We will say how do their cell walls differ from bacterial cell walls. So that true peptidoglycan, which I showed you a structure of, that exact structure is lacking in the cell walls of archaea. They have the most common cell wall is an S layer, which is a protein layer. Remember S layer we talked about? So S layer is the most common type of cell wall in archaea. They may even have a protein sheet external to your S layer. And inside, an S layer may be outside a membrane and separated by something called pseudomurine. Remember the word murine I told you is the word we use for peptidoglycan. So if it is a pseudomurine, it is a peptidoglycan-like structure, but it's not exactly the same. Pseudomurine may be the outermost layer, similar to gram-positive. So there are differences, a lot of differences in different archaea. So if you look at it, here are some of the cell envelopes of your archaea, where here in this first type, which is represented by Methanococcus, Halobacterium, or others, there is the plasma membrane covering your cytoplasm, and outside of it is nothing but the S layer. So the S layer becomes one of the cell wall component in a lot of these archaea. Then another representation is where outside of the S layer there is a protein sheath also. So that is methanospirillum is represented. The cell wall is like that. Whilst in this third one, which is the methanocercinia, they have S layer. S layer, as I said, is very common in most of the archaea, but they have a methanochondritin as a layer on the surface of the S layer. Whilst in your methanothermus and methanopyrus, they have the S layer, but under the S layer is what we call the pseudomurine. So that is a substitute for the murine, a different one. But again, it is not the true murine as you find in bacteria. And finally, there are some, such as the methanobacterium and the halococcus or so, they have a polysaccharide, just a plain polysaccharide layer as their cell wall. So as such, archaea have a lot of differences in their cell wall compositions. And here is the structure of pseudomurine. If you look at it, this is the one we had shown you as the structure for your regular murine, beta-1,4 linked NAM and NAG, the disaccharide. And then we have the lactic acid component, which allows it to get the chain of peptides, alanine, and so on. Whilst if you look at the pseudomurine, instead of the glucosamine is still there, but instead of muramic acid, this is a uronic acid. So this is a little different structure. And of course, there is the peptide side chain, but there is no NAM, as you would call it. And the linkage is also very different. So beta-1,4 linkage between the two sugar moieties in the backbone. Here it is beta-1,3 linkage. So what I'm trying to say that whatever dissolves, whatever enzyme or chemical that dissolves your bacterial cell wall may not have the same effect on the archaeal cell wall. Moving on to plasma membrane of bacteria now. Of course, this is an absolute requirement for all living organisms. Some bacteria also we have discovered now that not only do they have plasma membranes, but they have internal membrane system that will be found inside the cytoplasm or spanning the cytoplasm also. So we have discovered these recently to say, in the past we used to say there is only one cytoplasmic membrane that encompasses and we had never seen the internal membranes, but now we see it in a lot of photosynthetic bacteria or so. And this will be demonstrated in the next few slides. Okay. So what is the function of the plasma membrane generally? It has to cover the cytoplasm. So it is contained within this entity, which we call a cell. The main function of any plasma membrane, of course, is selective permeability. It allows certain compounds to come in, which is nutrients, and then it will allow some of the waste material that has to be dispelled out. So selective permeability both ways is the function of your plasma membrane. And a very important part of your plasma membrane in the bacteria is that they are the ones that interact with the external environment. So there will be a lot of receptors for chemical signals from the surrounding. A lot of transport systems are in the membrane, and even metabolic processes are located in the membrane. So what is the main metabolic process that we have to tell you about is, of course, your energy production. you realize in your eukaryotic cell, the energy production or the electron transport chain is in the mitochondrial membrane. But here in bacteria, since there are no mitochondria, the cytoplasmic membrane is the one where energy production occurs. So definitely, I would say for bacteria, their plasma membrane is a very important component for their survival. So what is it made up of? We know all membrane structures are composed of phospholipids. And phospholipids, of course, as we will look at them, they are, we have in the membrane bilayer, that means two layers of the phospholipids. And the phospholipids are usually represented as a head and two tails sticking out as this is one layer of your phospholipid, and this is the opposite, the second layer, which actually faces each other, so that the heads are on one side and the tails in both of them are hanging on the inside. We call them amphipathic lipids because they have polar heads. The heads are hydrophilic, whilst the tails, which are made up of fatty acids, they will be hydrophobic. So as such, the membrane is very hydrophobic in the middle, and on both the sides, inside and out, it is capable of water reactions occurring on that outside. Across the membrane, there are some proteins that are embedded or they are attached to the membrane. We generally kind of call them either peripheral proteins or integral proteins. Peripheral proteins are not going through the membrane. They usually are on the outside. I would say inside of the cell. This is the inside of the cell. So the peripheral proteins are stuck to the membrane through integral proteins that usually span the membrane, and they are very important. Okay. Kind of a general structure. Here is your phospholipid, which is, of course, phospholipids are composed of glycerol and ethanolamine, which is the polar end, which will be the charged end. And here we have the fatty acids as two tails sticking out. Remember, glycerol has 3 carbons. So one is taken up with the phosphate. One is the carbon here, and the other two, they are linked to your fatty acids. Okay. So as such, we talk about saturation levels of the membrane lipids. So membranes will have different saturations, meaning there could be unsaturated fatty acids which have a low melting point that could be present, and this will usually be found in bacteria that are psychrophiles. So what do I mean by psychrophiles? Well, this term you may have heard before. What is the property of a psychrophile?
They like living at lower temperatures.
They are able to live at low temperatures, and they may be in the ice-capped mountains or so. So in that case, unsaturated fatty acids will be found, okay, in psychrophiles. Whilst in your thermophilic bacteria, which can tolerate high temperatures, they will have more saturated fatty acid that will allow them, that will maintain their fluidity in the thermophilic environment, in the high temperature environment. So as such, these differences are seen based on which type of microbe you're looking at. So membrane lipids are different in different types of bacteria in terms of saturation of the fatty acids. And here we have also one more thing that I would like to mention is that bacterial membranes lack sterols. You know, eukaryotic cytoplasmic membranes have sterols in them. But bacteria do not have true sterols in them, but they have sterol-like molecules, which we label as hopanoids. So hopanoids are membrane lipids that are found in bacteria only, and we haven't found them anywhere else. This hopanoids actually is similar to the structure. Here is cholesterol, which is a steroid found in eukaryotes, and here is a similar structure you can see, but a little more branched or so. This is the hopanoid, which is found in bacteria. So these sterol molecules or sterol-like molecules stabilize your membrane And also important property that we've seen that these hopanoids are also found in petroleum, which tells you that petroleum was produced from some ancient bacteria. That's why the remnants of the hopanoids are found in petroleum. So an interesting discovery that we have. So how are the archaeal membrane different from your bacterial membrane? Archaeal membrane are composed of unique lipids. They have in the isoprene unit, which is a 5-carbon branched compound there, and their linkage to the glycerol is ether linkage rather than the ester linkage. So if you look at these structures that are given here, this is your bacterial or eukaryotic phospholipid. They have the same ester bond, whilst your archaeal ones are different from these two, and they have the ether linkage where the fatty acids are connected to the glycerol molecule. Okay, so major difference there. Also, in some archaeal membranes, you will have a monolayer. Usually, all our for the membranes of bacteria and even your eukarya are are bilayers. Here, you will find it to be a monolayer. So here you will see that these two fatty acids, you can see the fatty acids, they are coming out of one glycerol molecule, and here is the other membrane lipid, phospholipid here, but they are connected. So actually, there is no space as you would see in between when you have your bacterial membrane. So this would be considered as a monolayer rather than a bilayer. So they look like a bilayer, but they are the heads and the tails are connected together from one end to the other. So this will act as a monolayer. So here is a representation in picture here. You can see the tails hanging from the two phospholipids layers, and here the tails are connected. So this will appear like a monolayer for us. Okay, we move on to cytoplasmic structures, and this is an important, there is quite a few components in there that are different from your regular eukaryotic cell. Cytoskeleton is what we will discuss. So we will talk about intracytoplasmic membranes also that were new to us. Inclusions means storage material, and ribosomes, of course, are the place where protein synthesis occurs. So ribosomes inclusions are similar, or there is some commonalities in your bacterial and eukaryotic ones. And the last one, of course, is something that is unique to bacteria. We call it a nucleoid, which represents a nuclear area. That means the genetic material or DNA is a kind of in that area without a membrane covering it. We call it a nucleoid. And in your eukaryotes, we call it a nucleus, because there is a membrane-bound genetic material there. And of course, there will be plasmids also. These are extra DNA units that may be present in a lot of bacteria. Plasmids have also been found in some of the eukaryotic cells also. So we talk a little bit about the cytoskeleton. We know that commonly your eukaryotic cells have 3 major cytoskeletal elements, which we call tubulin, actin, or intermediate filament. So these terms are used for eukaryotic cytoskeletal elements. Tubulin, it's a protein, actin, which is also a protein, and your intermediate filament. So these are found in eukaryotic cells. So since we already knew that, whatever is similar to a tubulin, we call it tubulin homologue. So tubulin homologues are found in bacteria. They were discovered much later. And these are FTSZ. And this function of this protein, tubulin homologue in bacteria, is participation in cell division. And this has been widely observed in bacteria and archaea both. Okay? While there are other tubulin homologues that may be found, and we've looked at them in some bacteria, such as the tub A and the tub B. Their function is unknown in the bacteria, and these have been observed only in the Prostacobacter species thought to be encoded by eukaryotic tubulin gene obtained by horizontal gene transfer. So definitely it seems like some genetic material from the eukaryotic has passed on by horizontal gene transfer, and only this specific bacterium can produce this tub A and tub B. Um, Interesting. We should look at those papers also someday. But anyway, there is another tubulin homologue found in a bacillus, our common bacterium, and this is a tub Z. Tub Z is a possibly the function is to for plasmid segregation. Now, segregation is an important property that we have to think about, because you know that bacteria, when they have to divide, they elongate, and then a cell wall and cell membrane is laid in the middle, and that's why we call it binary fission. That means the two daughter cells will be of equal size. Now, when such a division is occurring, binary fission is occurring in bacteria, how we know that the DNA has already replicated and your nuclear material is already made into two copies by now. But what is since there is no spindle fibers or anything to pull the chromosomes apart, we always worry about how this division of the cell occurs, so that a representation of the nuclear material as well as the plasmids that are in the cytoplasm, how do they segregate? So now we are discovering certain specific proteins that assist in even the chromosome segregation as well as your plasmid segregation. So tub Z is considered possibly to have a function in plasmid segregation. And this is encoded on a large plasmid that is observed in the members of the genus Bacillus. Okay, so this is the rod-shaped bacterium we're talking about. Then there are some actin homologues, and one of the very interesting actin homologue is called MAMK. And this is definitely only found in magnetotactic species of bacteria. Have you heard of magnetotactic bacteria before? What is their, why are they called magnetotactic? Have you heard of them or no?
No, I personally
I
haven't.
haven't.
No.
You haven't? So these are the bacteria that can respond to magnetic fields. And so the earth has a magnetic field, and these microbes, very strangely, have magnetosomes in them. Magnetosomes will respond to the magnetic field. And these bacteria, I know a group of people who work in research, not on our campus, but they use magnetotactic bacteria. And if you have them in a liquid environment and you have a magnet under the petri dish, you can move the bacteria with moving your magnet. So it is kind of interesting that they have magnetosomes and they align themselves to the Earth's magnetic field. And I don't know why or whatever, but definitely this MAMK is an actin-like protein which helps in positioning of the magnetosomes. And we'll look at some of the pictures of the magnetosomes in the next few slides or so. Then we have a couple of actin homologues, which we call MREB or MBL. And this is found in most of the rod-shaped bacteria. And this actually is very important in determining the cell shape, and may also be involved in the chromosome segregation, or even to it localizes certain proteins also. So MREB and MBL have been worked out quite a bit. A lot of research has been done on these two actin homologues of the bacterial cytoplasmic structures. Then we also have another one which was discovered in some bacteria. This is PAR-M, and this is involved in plasmid segregation. And of course, this is plasmid encoded only. That means the genes for it are found on the plasmid. Then we have something that is homologous to intermediate filaments that is found in your eukaryotic cells. And this is, again, a very interesting one, which we will look at a little bit later. It's called CRES, crescentin protein. This crescentin protein is found in Caulobacteria crescentus. Now, this bacterium is one that has a curvature in the rod. So it's a curved bacterium, and this protein induces a curvature in the rod. If the mutant of CRES was found, this Caulobacteria would be a straight rod. Okay, we usually find the functions of any protein by finding mutants in them and see what properties lost. So finding mutants in these homologs allowed us to determine what their function is. But then there are some unique bacterial cytoskeletal proteins that are not found in, that have not been discovered in the eukaryotes. And one of them is MIND. MIND is, has been studied quite a bit, and it actually prevents the polymerization of FTSZ. Remember this FTSZ, which is found in most bacteria and archaea. This is actually a controlling factor, as I would say, in many rod-shaped bacteria, that MIND polymerization of FTSZ will not occur if MIND is not there. Then there's another one, which is a PAR-A, which is a very important one also. And this also allows segregation of chromosomes as well as plasmids. And this has been observed in many species, including your Vibrio and the Caulobacteria and the Thermus, all those very special ones also. So this is just a small list of it. We've been discovering all these cytoskeletal proteins in the bacteria now, and they are quite varied. So this is a very short list. There's many, many more. So now, just to go ahead and tell you that there are 3 that have been best studied, and these were the earliest ones that we had discovered, and we described them as FTSZ, MREB, and CRES, and of course I gave you the functions of these. So here is a